Okay, now we are in section 3, gene linkage and mapping. We learned that genes can be mapped to specific locations. Through these maps, you can identify the locations of genes on the chromosome, okay? And there is a scientist who called Morgan. He found out that there are some linked genes that inherited together, okay, on the chromosomes, okay? Now, during the meiosis, crossing over will happen between the two sister chromatids, between two chromosomes, and this will cause crossing over, so this will make diversity in our traits. Now, crossing over related to distances between genes. So the farthest are the genes, the more chance for a crossing over to happen, okay? While if the genes are near to each other, less chance for a crossover to happen. And the main important point you have to know here about linked genes, that they are the closer together two genes, they are more like to be inherited together. But not all the time these two linked genes inherited together. Okay? Now let's go to the section which is section. Now we finished chapter 7 till section 3. Let's go to section 4. We learned in this section that chromosomes, okay, and the genetics about humans can be studied, okay, and there are combination of methods used to study these the human genetics or human chromosomes. We said that we have autosomal genes disorders, 6 linked genes disorders. So we have autosomal genes disorders, يعني disorders that related to the autosomes, genes on the autosomes, هم من 1 to 22. وفي عندي disorders related to 6 linked genes disorder اللي هم 6 chromosomes. Okay? We learned before that the Punnett square is a method to know or to predict the phenotype from the genotype. Phenotype from the genotype. Now there is another method which is used, which is a pedigree. It's like a family tree. It is used for tracing genes in a family. يعني to trace or predict the genotype من الphenotype. It's opposite to the Punnett square. Now these phenotypes are used to interfere genotypes on a pedigree. We have this pedigree, we can make this pedigree for all disorders, for autosomal genes and for 6 linked genes. The difference between two is the carrier in both of them. In the autosomal gene disorders, male and female could be carriers, but in 6 linked disorders, there will be carrier only in the female part. Why? Because as we learned, the male has only 1 copy of each 6 chromosome, so all the genes will be, the alleles will be expressed on the genes. Okay, now there is another important thing you should know. The square represents the male, the circle represents the female, unshaded shape represents a person that doesn't have the phenotype or the disorder or the trait, but shaded, that means that the person has the phenotype or the disorder or the trait. Okay, if it's half-half, that means the person is carrier. There is another method to study the genetics or your chromosomes is the karyotype. Karyotype show you the picture of chromosomes in the cell, and if there is any abnormality, you can identify it. Now, karyotype can show changes in the chromosomes if there is any deletion or if there is a duplication or extra chromosomes number or large change in the chromosome. All right? So now we finished chapter 7. We will start in the next session with chapter 8. Thank you.